Good morning, everybody, and happy Friday. Robert and I have not seen all of you in a very long time, huh, Robert?
Mm-hmm.
Feels like forever since we've done a live stream. Well, thank you for joining us today on this Friday for our 2-Bit Circus Foundation Hour of Playful Learning. So we have a fun activity we're going to do today, don't we, Robert?
Mm-hmm.
You want to tell them a little bit about the stuff that we have on the table here?
We have onions,
Mm-hmm.
salt, different kinds of salts, water, leaves.
Awesome. So we have a bunch of variety of different objects that we found around our house and outside, right?
Mm-hmm.
And we have a question that we're going to do today. We're going to try to determine whether things are living, if they're alive,
Or they're non-living.
and if they're non-living, they're not alive, right?
Mm-hmm.
Awesome. So what do you think, Robert, about the stuff that we have on the table? Does everything look like it's alive?
Mm-mm.
Nothing looks like it's alive, huh? So we're gonna have...
I think 2 objects to me look alive
Mhm.
so far.
Which ones?
The leaves and the onion, but if you take off the leaves off its branch, it's dead.
Oh, okay. So, yeah, maybe it could die, right?
Mm-hmm.
Like it's gonna die over time because we removed it from its source where it gets its energy from. But at one point, maybe it was alive, right?
Mm-hmm.
And if we looked at the material list, there was actually another material on there that's not on the table that I put on there. And that was a human. So Robert is going to be the human sample for today, too. So we'll see if Robert's alive or not.
If I'm talking, or if I am doing this,
Mhm.
or if I could play video games, then I would be alive.
Okay, cool. But does an onion play video games? When's the last time you saw an onion play a video game?
Yeah.
But you said the onion was
But
alive.
I eat also. I eat donuts and different stuff.
So you eat different stuff, so that makes you alive, right? And of course, our doggy Cole, he doesn't play video games, does he? But he's still alive.
Mm-hmm.
So how can we determine whether this stuff is alive or not? What's something that we could do to determine whether these things are alive? Or were alive at some point?
Until you killed them.
Until I killed them, I pulled the leaf off the branch.
Yes, you killed him.
Okay.
You murdered.
Okay. So hey buddy, look, I have a little thing I set up here on our little cutting board. I made 2 signs. One of the signs says living.
And non-living.
So it says living on there.
Non-living.
And non-living on the other one. Awesome.
Non-living.
Look, Robert's getting so professional with this, huh? Awesome. Alright, so Robert's going to do a task for us, and this is going to be his hypothesis to his experiment. He's going to sort out the items that we have in the center here. He's going to sort them out onto 2 teams, 2 sides. He's going to tell us whether he thinks the item is living. I did that backwards, huh? He's going to decide whether the thing is living or non-living. Alright, so Robert, you ready to begin?
Switch to this side because you said I'm the object, so I will go on one side.
Okay, so we have to switch sides because Robert says he is alive and he is going to be one of our test samples today. So he went to the living side. So one thing is over there already. Robert is living. Okay, the plant leaves are living. Robert said the onion he thinks is living. Okay. Anything else living that you're going to put on this side? Awesome. Interesting. All right. So, Robert, can you stand up straight today? All right. Why don't you lift up each item, tell them what it is, and tell them whether you said it was living or non-living.
Leaves.
Okay.
Living.
Show it to them. Show it to the camera so everybody could see. So leaves are living according to Robert. Okay. What else?
Onion.
The onion is also alive.
Human.
Human. So we have 3 things that are alive, right? Leaves, onions, and humans. Okay. And now what do we have that is non-living?
sugar.
Okay, so we have a container here. It says sugar on there. We have labels on all our things.
A little wine cap.
A wine cap. And do you know what that material is called? How do you know this stuff is a wine cap? You see, Mom?
Because I see you in parties drink it.
Oh.
You use a little wine twister to drain it off.
Wow, this guy. Okay, awesome. So we have a little cork here, cork material. This is an unused cork, so it's not from a wine bottle, but it is for science, right? So it's clean, it doesn't have anything on it. Okay, awesome. What else is there?
Water.
Water. Okay, so we have a container of distilled water, purified water in here. It's labeled water. What else?
Pepper.
Ah, close. I don't think you want to eat that though, or put that on your food. And I missed the label.
Dirt.
It's dirt. Okay, so we have a container of dirt here. And let me label that because I actually forgot to label it before we started today. So, okay, I can't write on twisted glass, but that's okay. Bent glass. But there we go. Yes, he is a cute little superstar. And we have salt. Awesome. So we have our items that we sorted out here for us today. Again, we sorted them out into two categories, living and non-living. And so Robert just made this hypothesis based on prior knowledge that he knows about things. And Robert learns a lot about plants and stuff, right? And humans and living and non-living things at school. So he might have an idea here. So we have these things sorted out. Now, what I have for us as well, and Robert, can you scoot over a little bit so I can reach that mouse over there? We have a special setup for us today.
Why is glitter on the table?
From our other projects that we were doing. Right?
Red dot, red dot.
Red dot. Okay, let's not get distracted though. So we have living and non-living things here. And we have a microscope that I don't know if you can see. You can see it right here, a little bit on camera. And we have a camera hooked up to the microscope to share with all of you as we do our experiment today. And for the experiment, what we're going to do is we're going to take little... Yes, go ahead, Robert. Tell them.
We're gonna take a little piece off and then put it inside here and then put it in the microscope. See if it's alive.
Awesome. And how do you know if something's going to be alive after we put it under the microscope?
Because if it doesn't have cells, it's not alive.
Oh, so you learned something about cells already, right? Did you learn that at school?
Mm-hmm.
Okay. Wow, you go to a good school. They taught you about cells already? Awesome. So we are going to be learning about see if things have cells by putting them under a microscope. And so you can see here that we have a fancy microscope. But if you don't have a fancy microscope like this at home, and you don't really want to spend a lot of money on a fancy microscope, the little kid microscopes that you could get for around $20 or so will allow you to do this lab as well. You don't need something fancy with electricity and light. One of the basic ones with just a little mirror will work under there. Because Robert, you know they discovered about living and non-living things way before they discovered about electricity? Did you know that? They knew about living and non-living things and what comprises them before they even had electricity.
Wait, maybe the first thing they discovered living was them.
Maybe they did, because you have to be alive to make discoveries, right?
Mm-hmm.
Awesome. And so, yeah, you could use a little basic microscope. It doesn't have to have electricity. The old original microscope just had a little mirror where you take a little bit of sunlight or ambient light and it'll reflect it through the slide, and that will allow you to do the same thing on a budget, right?
Mm-hmm.
Awesome. Let's read some of the comments here before we get started. Keep us on our toes, that's okay. Yes, let's switch it up a little bit. Less talking from Michael and more talking from Robert. Okay, awesome. So we are going to switch over to a view here so you can see, hey, you can see a couple views. Awesome, so you'll be able to see us, Robert and I. You'll be able to see our tabletop over here on the side. And then this one over here, the big picture, is an image of the microscope. And right now there is no slide under there, so you're just looking at the diffused light coming through the hole. No, I have the light on here. My assistant off camera was saying that it might be the light from there, which is true too. If I were to turn off the light, again, you could use just ambient light to see what's on the slides. So this is our setup today, so we could do our experiment. And we're gonna let Robert prepare slides, because he's learning how to do that, right? How to get the slides ready to go. And then of course I will help him. But we can start with whatever object on the table Robert wants to start with.
I want to start with the onion.
Okay, cool. You want to start with the onion. So do you want help cutting that onion? Because I don't think that onion's going to fit on the slide like that, do you? So how do we have to prepare our slide to put it under the microscope? Okay, we can look at that. You want to take a little bit of skin and look at that under there too? Do you call that skin? I don't know. What do you call the outside of an onion? The outside layer. There we go. Awesome, Robert. You want to come on the other side now? Because you're going to be a scientist right now, and then later you'll be part of our experiment as well.
And then can you do it to me?
Yeah, well,
Because I'm an object.
yes, and you're an object in this experiment. So after we're done experimenting with these objects we have on the table, we're going to try to find a way that we could put Robert, or a part of Robert, under that microscope. How does that sound?
How about you make it go up, 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 up?
I don't know if it goes that high. Okay, so let's go ahead. I'm going to help you slide this slide under here. So we get a nice image. Hopefully we can get a nice image. And then we got to focus the microscope.
I see like chip layers. It looks like a planet.
I saw like a little bit on there, huh? Something. So it's kind of hard because this isn't flat. Well, let's see what we see under here.
It's living.
Can you see it, Robert? And you can look at this closer view here too. Awesome. Hey May, they could see you off camera hiding there. You might as well just say hello.
Hello, everybody.
So it looks like my stream got taken over. We've been hacked. The science teacher, the middle school biology teacher, was trying to show us how to use a microscope properly, huh? So tell us what you're doing there.
Okay, so to use a microscope without breaking your slide,
Sure, we're listening, okay?
you need to make sure that you start at the lowest objective lens. These are called
Oh.
objective lenses.
So on the microscope, I don't know if you can see on the picture there, you might be in the top right corner, there are these 3 lenses here. And what our teacher is telling us is that we could actually change them around to change the magnification, right? So we could get a outside look, and then we could zoom in, and zoom in, and zoom in, huh?
So when you're at the lowest objective, you can use the big knob, which is the coarse objective knob, the coarse knob,
Can we see it?
which moves your stage up and down like really fast.
Oh, that's how it works. I was just over here moving it around till I saw something cool. Was that what you were doing, Robert? Awesome. So let's look at that picture. You could either look at the camera here, or you could look at it on our screen here. And can you see anything in that picture?
Okay. And like Michael mentioned earlier, it's not a flat sample, so we don't have just one plane we're trying to focus on. It's like multiple planes. So we start to see something, right? Robert, do you
Robert,
start
look
to see
here.
something?
We'll think about that later. Can you see on that screen there? What do you see?
Little rectangles.
You see like little tiny rectangles there, huh?
It looks like rocks.
Okay, they kind of look like rocks, right? But you see a pattern in there, don't you? And you see a pattern of a bunch of little tiny rectangles that are like touching each other, huh? And what do you think those little tiny rectangles are?
The cells.
Yes, they are the cells.
So this is alive. I'm right.
Okay, so onions have cells. So they're they're alive, and Robert's doing his happy dance, his victory dance, because he got the first one right. Awesome.
Yay.
And Robert, do you know who discovered or who invented the microscope?
Albert Einstein.
Yeah, this is way, way long ago before Albert Einstein.
Michael Faraday.
Before Michael Faraday, you're getting closer though. I'm so impressed of all these scientists' names you know. I'll give you a hint, Robert. Robert Lusk discovered it. So actually, you remember that scientist we talked about like a long time ago? I told you there was this guy named Robert Hooke.
Oh yeah.
Yeah, Robert Hooke actually was the first person to invent a microscope, a microscope like this, and he was the first one to observe cells. And do you know how he came up with the name for a cell?
Because of how it looks.
Because of how it looked, and it looked like something that he had seen in real life. So he...
A seed.
A seed?
Cells.
A
Think
cell.
about cells.
So Robert, in Robert Hooke's days, they had these big churches, and in the churches they used to have these rooms where all the monks used to stay. And they used to call those rooms that were in a pattern of little rectangles, they used to call that a cell. So they used to live in churches in little tiny cells. So that's how he
Like
came
a
up.
jail cell?
Like a jail cell, but they weren't locked in there, okay?
Yay!
But that is exactly how they came up with the name cell, okay? From a scientist named Robert Hooke. And now I know I'm getting all off, but I always like telling this story. Do you know that we don't know what Robert Hooke actually looks like? He helped us look at so many different things, but there's actually no pictures of Robert Hooke. Yeah. There was one picture at the Royal Society in England where he was the head of the Royal Society because he was an amazing scientist. But him and another scientist named Isaac Newton, they didn't like each other. They had a lot of drama and fights. And so it's said that after Robert Hooke passed away and Isaac Newton took over his position, they think that Isaac Newton might have destroyed all the pictures of Robert Hooke. That wasn't very nice, was it? But that means we'd have no pictures of what Robert Hooke looks like. That's kind of a crazy story, huh? Awesome. Well, anyways, that was a nice sidebar to teach us a little bit of history of science and the people behind it. But we should get back to our little story here about Robert Hooke and the microscope and the cells that we see under there. So Robert was correct. An onion is alive. Now, before we move on, I would like to actually cut open the onion.
Ooh.
And we could try to get a smaller layer in here that's flat. You don't know about onions yet, huh? You don't usually eat onions.
And they smell gross.
They do smell gross, and even more than that, if you get too close of them, they release like a chemical that makes your eyes water. So you're gonna start crying and your eyes are burning a little bit because we got fresh onion here. So, awesome.
Ew,
All
what?
right. Robert, get up here. Hey, let's not get up here. Awesome. Is that a thin enough layer?
Yeah, you just gotta do it. I would do here.
I want to figure this out. Here, I got a layer. I got a thin layer. We're using this one. We're using this one. I have a thin layer here.
Okay, there's
All right, Robert. Do you want to touch it and get the onion smell all over you? It's okay. We're a family that doesn't like onions. So that's why we're chopping it up and using it for science only. That way I don't have to eat it later, huh?
How about we chop all the onions up?
Awesome.
Oh, but
So,
Robert loves Funyuns, right?
yeah.
But they don't stink like that.
Yes, they do.
No, they don't.
Yuck.
They smell great.
Okay,
All
Robert.
right. So what we did here, I don't know if you were able to see, maybe you were, but it's kind of hard to see something that's clear and then something so thin. But we put a little layer on top of the onion, on top of the slide. We put a little layer of onion on top of the slide, and then we put a cover on top of the onion, so that way it doesn't move around, and we make sure that it's nice and flat. Awesome, so we're gonna go back over here, Robert, and let's see what we see. Let Robert do that. Come over here and you could change the coursing on the microscope.
So come here.
Scoop. Ugh.
So I look. Have a seat this way.
And then you're gonna move this back and forth.
So I lowered the stage.
Oh, the stage
Use
is
the
on.
clips to clip this secured so it doesn't move. Okay. Let's make sure.
Yes, Gonzalo. E.L. Gonzalo.
So now-
Some hair follicles would be pretty interesting to use as well.
Use your
Maybe later, Robert, we could look at some hair follicles of yours too. You want to do that?
This one, baby.
The big one, the
The
big one.
big knob is gonna help
That is
you
a great suggestion.
move
We'll
up
look
the
at that
hair
as
quickly.
well.
Keep going. Keep going until you see something.
Yes, I think it's really awesome that you could show the microscope like this. In the old days, you used to have to look through the eye lens, but nowadays you could hook up. We have a device here. I don't know if
It
you can
looks
see
like
it
a
too well.
bridge.
That you replace the object on the top, and I was able to connect my phone, so that way I could look through the camera on the
It
phone,
looks
and you
like
could
a
actually take
bridge.
pictures and videos of things that you see under the microscope as well. So that's a pretty cool way that you could detail your experiments with the microscope. Some of us more so than others. Malin over here is a professional biologist. She knows how to use a microscope very well. Me with my engineering background, I just like to pretend I know. No, but they are definitely really fun to use. And they're simple to use for everybody. Once you just learn, the focusing is really the main issue. And then trying to get that through there.
Right. The problem is having to look through the eyepiece, and you have it focused for your students, but they have different what do you call it? Vision, right?
Oh,
So their vision's different. That's just water.
Oh,
that was, you put water in there?
No, it's just from the release
The
of
onion.
the onion.
There wasn't
So
that
you
much.
can see it here. Normally in class we use iodine and we dye the cell wall so we can see it clear. And you can kind of see those ridges there. That's the cell wall. I'm gonna flip it over to a lower,
There.
and you
That's-
can see a lot of them here.
Yes. You see that, Robert? Look at that shape. You see all those cool shapes in there? They're like all elongated. Not all
I
of
like
them
the
are.
other one when it was like my shoes, how they look on the bottom.
Yeah. How is that
Oh,
a
you
different
have
shape?
honeycomb.
He has honeycomb structures on the bottom of his shoes that kind of look like cells, huh?
From the other one, when it was lit.
That's water.
Yeah, that's the water that got splashed on by the release. of the
Awesome.
onion.
So you could see there, you know what helps too?
The diaphragm,
Changing the light contrast there.
the amount of light that comes in through
Yeah.
the diaphragm.
So you can kind of see the structures there better. Robert, look at that.
Robert, like,
You see the structures? So you could see the little cells in the onion. So you were right. Onions are alive, right? And where do onions come from?
what's an onion? Where does it come from?
The ground.
The ground.
And it's like a plant, right? It's a root. Okay. Awesome. What's our next one that we could test out here?
Which one do you want to see?
You want to
From
look at the
living or not
leaf?
living?
Or you want something else from the
I like this leaf.
that leaf? All right. So we could try a couple things here. The leaves are challenging too, to try to get a thin layer of the leaf. I don't know if I'll be able to separate it. You You can just put the leaf under the microscope, but it's better if you get like a little sample layer. Some leaves are thicker than others, which allow you to separate the layers. And if you have more precise fingers and smaller fingers than I do, you might have better luck. Robert, are you able to separate those layers of the leaf?
Try.
Can you try? You have really tiny fingers. You might be having better luck than I do. And some of this stuff takes a lot of patience to do, and maybe this is why I never became a biologist. I went the physical science route. Okay, we could see if that sample works. Which one? We got a clean slide here.
Mm-hmm.
We have a clean slide. Yes, exactly. And then Ian, back in our days, we used to wait in line to see what was under the microscope that the teacher was showing us. Now you could just display that for the world, kind of like we are doing here today. And then Robert, remember, so you always start with the lowest magnification.
Start with the lowest, and then you bring the stage up.
And maybe we change the diaphragm with the light here.
Sorry, you might need more
Because
light.
it's thicker.
Look at that.
Oh, look, Robert.
Right there. You see that?
Oh, wow.
Okay,
So, what
that's
do you
cool.
see in there, Robert?
Alright, leave it. What do you see?
Cells.
Cells.
Those are cells.
You think those are cells? Cool.
Alright, now we use the little one.
So we're going to zoom in more.
The fine adjustment, the little one. Because you don't want to bump the slide on the...
You already
Yeah,
moved
there we
it.
go. Okay, there. Okay, now use the little one to focus more. If it's getting too blurry, then you're going the opposite direction.
Whoa.
Okay,
What?
so I think
I
that
think
was
you
a good
passed
spot.
it. You
I think
passed
that was a
it.
good
You go
spot.
slow.
Alright, about there. Alright. Now we're gonna bring the higher magnification. Hopefully we don't move our leaf. Okay, and then use the little one.
Alright, so I know the image looks kind of blurry here, but right now they're focusing in at the different magnifications.
Not too much. This is super tiny. Tiny, tiny movements. Oh,
Andy and Ian, you can buy simple microscopes off of Amazon. They have a huge different variety that you could look at. You could have expensive microscopes that are a couple $100s, or they even have more affordable ones that I would suggest looking into getting that are around $20 to $40 range, and you could do a lot of things with those little basic ones that I'm doing here. And usually the magnification capabilities are exactly the same as the one that I have on video right now. So anybody who's watching this who really wants to do the work that we're doing and even do additional experiments with other things that you could find around your house, you can get a microscope off of the internet or in the stores. I think most stores sell a basic kit, like in the toy section. Those toy microscopes can work as long as you can get light to come through the slide. Right? And then they also have stereo microscopes, which are a little bit different, which shine light from the top surface, and that allows you to look at objects like a leaf and not necessarily see at the cells, but see a closer zoomed in look of the structure. And that's really helpful for objects that are not transparent. Like if you find a bug, Robert was whispering in my ear earlier, he wanted to look at an ant under there. And without dissecting the ant and taking apart the layers of the ant, it would be really hard to use on this microscope. So you would need a different type of microscope to look in and see the structures of it.
Why don't we take it apart?
Yeah. How are you doing there, Malin? Are you able to find the cells?
Really
and
hard.
Yeah. Well, we could just see them with that, no? Awesome. All right, Robert. So you think that plant leaf is alive, or you think it's non-living? Living or non-living? I think there were cells in there too, huh? Yeah?
Let's try the green one.
All
quick
right.
while you talk.
Well, let's look Well, let's move on because we have a bunch of stuff we have to get done.
Okay. Let's see. This
Oh,
is
look
like
at that one real quick, Robert. That one's easier to see.
Yeah.
That one's
You see the
great.
structure.
Let me see the structure.
It's because it's dark.
Yeah, I think because maybe this leaf, I don't know.
It's too light.
It's maybe it's too thick. You couldn't
I think
see.
the yellow.
The
And it's
yellow.
the thickness as
Yeah.
well.
Awesome. So we're getting zoomed in more and more there. And as Malin is finishing up with that, I'll start prepping the slide for our next object that Robert is going to test to see if it's alive, living, or non-living. Robert, which object do you want to look at next?
Okay, Robert, you see all.
Do you want to look at some of this? Right, and this is sugar. Let's see if I can pick some of the sugar up. I should have brought a spoon. This doesn't work unless you have liquid in it. Alright, let's just be messy.
Dump it.
That's what I'm going to do. I'm just going to dump all the sugar. Right, we're not so fast. Oh, that's a lot of sugar. Don't eat it though, because this is an experiment, right? Up. I just dumped all the sugar onto the breadboard here, the cutting board. So let's move our sugar into the center. Awesome. Hey Malin, should I add water to the sugar, or should I just make a dry slide?
You do a dry slide.
Now, what's the benefit of adding water to slides like that?
You break things apart.
It separates things,
Mm-hmm.
suspends things. All right. I don't know if I'm doing this right, but here you go.
Okay, let's move this one out of the way.
I thought they were supposed to like stick, no? That's with the water. So technically I don't even need a
You
slide
don't
cover.
need a cover.
Alright.
Hey, Robert.
Okay, Robert, look here. Look what
do
we
you
have
see
under
there?
here. So now under the microscope,
It looks
we
like
have
ice.
It
sugar.
looks like ice.
And I think the image should be popping up for you all soon. Awesome. There we go. Want to make sure the
It
cameras
looks
are working.
like blocks of ice.
Yeah, look at that. It looks like little cubes, right?
Mm-hmm.
Do you see any cells in there though?
No.
No. I don't see any cells either.
So it's not living.
Non-living. Sugar is non-living.
Another point.
Awesome. So Robert's been correct on everything so far, huh? All right, let's prep the next slide. What are we going to do next, Robert? You want to do this one next?
With
Cork.
sugar.
Well, you can't mix the samples, right? Because then we won't know which is which.
How about we do that first and then we put sugar on?
Okay. After we look at it, we can. So if you look at the sugar, you could just see the crystalline structure of the sugars, but you can't see any cells in there. So that's a good indicator that sugar is not alive. Good thing it's not going to jump out at us. The next thing that Robert wants to look at is that cork. That cork. So what I'm trying to do again, because cork is not transparent, it won't work very well with this microscope unless you get a really thin slice. So I'm trying to get a really thin slice here that the light will be able to see through. And I was also trying to get the piece to be relatively flat. Now I wonder if this piece is still going to be too thick or not. But let's see, Robert. What do you think? This one's going to be interesting, Robert. Let's look at this. Let's look at this.
It looks like Mars.
It looks like Mars, okay, because it has the colors in there, the reds and browns, right? And look, Robert, let's look. As that microscope is getting fixed and focused. Okay, can you see anything?
It looks like a sunset and mountains.
Sunset mountains.
Are you able to
It
see?
looks like a shooting star in outer space there. Next to Mars.
I wanted to get more light. Is that the diaphragm one? Cool. So we're adjusting the microscope now, trying to see if we could get focus onto the cork. And I don't think we're getting a really clear image here.
We need
We need
a nicer cut.
a nicer, thinner cut piece.
A smoother cut.
Yes, it does have a space feel in there. Alright, let's see. Let's try to get a thinner layer. Having like a scalpel or something or biology tools would be better, but instead I have my tools.
We have engineering tools.
My engineering
to do biology work.
tools to do biology work. That's how I roll. Alright. Watch out, Robert. Sit back. Sit back. Back. Hey. Is that in your mouth? No. Okay, here. Thought it was in your mouth.
No, he loves sugar.
Don't put stuff in your mouth while you're doing labs. That's yucky. You get
It's one
sick
of the lab
that way.
rules. Just don't taste anything unless your teacher tells you.
Don't taste anything.
Alright, let's see if this one works.
It looks like American
Oh,
flag.
this one looks like
Oh,
it works.
I think I see something. Robert, did you see that? Look. That big piece was good, man, on the side
The
there.
bigger one.
Yeah.
That one looks like
Right there. That part
I
there.
can't let me bring it.
So
I hate
the
how like it's inverted, and
it's not
always-
only inverted, but also the camera is shifted and rotated.
So then
it's
it's like
inverted
a
like
weird,
twice.
yeah.
So even your normal inversion isn't gonna be. Let's play with
Usually I
that.
tell my students move it opposite.
the
If you need to move it up, then
It
move
looks
it
like
down.
the moon.
Yeah. Let's see.
Half of
For
the
you.
moon.
I'm trying to. Let's
I think that
change.
should be fine.
Changing
It
two
looks
things
like
at
a
once.
crescent.
Robert, can you see in there?
Give it more light.
Much more light.
It looks like that's the water and that's the earth.
Yeah, Robert, look in there though, really close. You could kind of see. It might be easier for you to see on here.
It looks like the world's attacked.
Hey, you're swimming so fast. You're not letting me see. So I was able to see, before the objective lens was moved, that I saw some shapes and patterns in that cork. I don't know if you saw that, Robert. Did you see that?
Now I see
More. We're almost seeing it.
Here, you do this part.
See, I'm messing with the fine one too. I just moved the big one. Of course, you'll be able to see it. I am trying to dissect this cork into a thinner piece right now. I don't know if I can do it. Yeah, let's try doing this. Clean cut. Well, kind of.
Is it?
Alright, let's see. Let's go over, Robert. Let me take a look at that. We're gonna try to get a cleaner cut for you. That's what Malin is going to do. So we're switching roles. And I am going to go back to the microscope here and see if I can see anything. And I could see something. I don't know if the glare is too much with the camera. Robert, come look at this. So if you look here, Robert, look through here. It's kind of glaring on the camera bad. Hold on.
No. It's dead.
It's dead. Are you sure? What pattern did you see in there?
A quart bottle's not alive.
A cork bottle's not alive. Robert, sit down. Thank you. Don't bump the microscope. Alright. So I saw in there a pattern, Robert. I could see it right there with that clean cut. Look at that. Robert, do you see that pattern? You can look there, you can even look on that screen. Can you see the pattern?
Yeah.
Yeah, so what does that look like? They look like cells, don't they?
No, but a quart bottle's not living.
A cork bottle's not living, but maybe that's something we should think about. Where does cork come from?
Onions.
I think that's pretty clear.
Bottom. Well, on your right.
On the right side, remember, don't spill water, please. On the right side there, you can see a little bit of a pattern from the cork. And so the cork actually does have cells. I love like doing that with the light. The cork does have cells. So Robert, where does cork come from? It has cells, so the cork had to be living. It's probably not alive now, because it's like dried out and dead. But it was alive at some point. So where does cork come from? That's a tricky question. It's okay. It actually comes from plants. So you use tree bark to make cork. It's dried out bark. So if you look at cork, you can actually see the cell structure of what's left of the cells before before the cells in the cork died. So at one point, the cork was actually alive. And you can see on the screen now a bunch of those cell structures in the cork. So Robert, this is Robert's sad dance, he said. Because this was the first one he was wrong on. So that's okay. That's why we do science, right? Our hypotheses don't always have to be correct. But we have to make observations, and we have to use that observations and data to inform our future decisions. Come on up here, Robert. We got a couple more things to look at. And we can't forget to do the experiment. You got to remind me, we still have one thing on the living side that you have to test. We still have to test the human. Okay? So let's finish up with this stuff here so we can get to the human test. I think that's going to be the funnest test to do.
Humans are alive.
Humans, I don't know. I'm not sure. We got to check
They
to make
have
sure you're alive.
brains.
Does the brain make you alive?
They have hearts.
Yeah.
What does the heart help with?
Yeah, where does the heart, what is the heart made out of, Robert?
Blood and cells.
Okay, so it's made out of tissue, right? And tissue is made out of cells, right? So you have this structure of things being built. A heart is an organ. It's made out of tissue. And tissue is made out of cells. And then cells are made out of some little thing that we might discover later. Awesome. So we have our dirt from our backyard that we are going to look at now. And focus in here. See if we see anything.
Yeah, bring it up. Ooh, Robert, look at that.
So that's what our dirt in our backyard looks like. And I don't see anything like cell pattern structures in there, do you?
So it's not alive.
It's not alive. Let's try to zoom in closer just to double check. You know, I thought it would be funny if there was like a bunch of like single cell bacteria or something in our dirt.
Because they might need a little bit of water.
Yeah. That's okay. We could conclude that it's not alive, right, Robert? Are you okay? Are you good confirming that our dirt is not alive?
Mm-hmm.
Okay, so you gotta do your happy dance now. Awesome, there's your happy dance. Do we need it? We'll add water here to test the water with the dirt.
That makes
So that way
it
we can look at
mud.
the dead. 'Cause I
That's
don't think we're gonna
about-
see much under the water alone. I'm gonna just
That's
put it
its
aside.
last evolution. Mud
Mud.
is dirt's evolution.
No, hold my hand. I'm gonna put a top on. What do you call those tops, by the way, so I can stop calling them tops?
This one's like
We just
slide
want
covers.
slide covers. Awesome.
I'm like, I just want like.
I might as well just call them tops.
Right, it's such a simple name.
I thought it was
Tops,
gonna
like
be like a
box tops?
fancy word. I don't
Nope.
know if we're gonna. Alright.
Ooh,
Not too fancy.
So we just added a little bit of water to the dirt here. To separate the dirt out a bit. And I'm pretty comfortable saying that our dirt is not alive. I don't see any cells in there, Robert, or any cellular structure. Do you?
yeah, I do my own project.
In a minute. We want to do this other stuff real quick. Cool. We're gonna test you in a minute.
Which one are we missing?
How? How are we gonna test you? Uh-oh, it looks like I lost my comment feed.
How about you put my hand there?
Oh.
Okay, we could try that. We could experiment, see if that works. All right, Robert, so happy dance. You got this one right, too. Dirt and water are both not alive.
Da, 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 da.
So how many did you get right so far, Robert?
Mm. 1, 2, 3, 4.
And then the sugar, right?
Mhm.
5. So you got 5 right in your experiment, and 1 that wasn't correct, right? And which one was that?
Cork.
Cork. And so now if I would ask you, is cork alive?
Yes.
Yes, it was alive, right? It's dead now, but at some point it was alive, and that's because it came from trees. All right? All right, let's look at salt. We have a nice slide here.
Yeah.
And then we're going to do the funnest one. We're going to determine whether Robert, which is, Robert, you're a human, right? We got to see if humans are alive or not. I don't know. I don't know. Are humans alive? I think Robert's getting worried now. Alright, we're going to see if you're alive in a minute. Alright, so the one we're doing now, which is the last object we have on the table here, is the salt. So let's go ahead and put salt under
It's not alive.
the microscope. Robert's prediction was that salt is not alive. So here we have a pretty good image of a salt crystal. Has a unique structure. Each piece of salt, I'm sure, has a little bit variation in it. But it doesn't look like there's any patterns of cells or anything in there. Huh. So were you right about this one, Robert? Alright, is salt alive? Awesome. So, Robert has only missed one, which is the cork, which is a tricky one so far. Now, the biggest one we're going to check right now is this sample. We're trying to find a way that we could put Robert on a slide. So, Robert's putting his hand under the microscope right now. I don't think your hand allowed light to go through it. So, I don't think that microscope is going to work for that. Maybe if we had a stereo microscope, right? I always get them confused and I'm like thinking quickly right now. I want to make sure. Stereo microscope, so big one. Compound microscope. Awesome. So what we're going to do is we're going to do an experiment. We could test Robert's hair too, but I think this one is cool. We're going to take a toothpick, a clean toothpick. These actually are nice toothpicks. They don't actually have points on them. Are these special biologist toothpicks to do this experiment?
Yes, those are the ones I use.
Awesome. So these are safe toothpicks
They're
to do
flat.
this with. They're flat tipped. You could do it with a pointed tip toothpick as well. Just be careful that you don't like stab yourself with them. Okay? And what we're gonna do, Robert, is I'm gonna take a sample from inside your mouth. Okay? And we're gonna check to see if there's cells inside the tissue of your mouth. You ready to do that? Okay, say ah, go ah. All right, let's see here. How's that feel, Robert? Feels funny? All right, let's check. Make sure you're alive in there. All right, and now I'm putting Robert's like slimy, yucky stuff
You put it in an R.
that I got from his mouth there. Should I put a slide cover in there? Well,
If you're not gonna diet.
I'm gonna do a couple things. I'm gonna first look at his yuckiness. I could see it, Robert. Can you see all that yuckiness?
Yes, it looks like
Ew.
an R.
Disgusting. So we're gonna put this under here, and we're gonna see what we see. And now remember, these aren't dyed or any color, so it's just clear right now. So maybe or maybe not will we be able to see anything.
You gotta put it way higher.
It doesn't go higher.
Oh, that's it?
That's why.
Oh.
That focus is all out because you're messing with the fine enough. Alright, so I'm starting to be able to see the structure of some stuff, but it is still hard to tell what it is. Ew. Maybe less light through here?
Oh, there we go. Look.
Up.
Are those cells?
So I see yucky stuff that came out of Robert's mouth here. And maybe they're cells. We want to take a closer look in there, huh? Why don't you come over here between us so that way you can see the camera better, the closer camera. Okay. Ew.
They are cells.
They are cells. And these are actually really good cells. You could actually see stuff in them. Like maybe there's parts inside of a cell. Can you see that on that camera? I think you can a little bit on that part. And Robert, look, I see in there, I'm going to use the other one real quick. I see, like you see those dark points in the cells? I wonder what that stuff is. Okay, sit down for a minute because I'm going to move the slide. I don't want it to drop. So you can see that I think Robert is alive. I see cells in there. Now something you could do to the cheek cells, that's what we took from Robert's mouth with cells from his cheek. You could actually dye them with a dye called methyl blue, and that will help you see them. But I wanted to experiment whether I could, be careful with your cheek cell sample, I want to see if you could use simple household products to dye the cells as well. So a conversation we had was whether food coloring, normal food coloring that you have at your house, right, for baking and doing all sorts of stuff like that, be careful with the sample, can work. So I am going to try to find a way to get some food coloring on here, trying to see what color I want to use. But I don't want a lot, just a little bit. Okay, maybe a lot. The good thing is I don't think we'll ever run out of sample if I mess this up. Because Robert's right here, so I could just keep taking new samples from his mouth, right?
And then Robert and Ian said, "Congratulations, Robert, you're alive."
Congratulations, we have definitive evidence now that Robert is alive. Alright, so I put a
That
little
looks
bit of
like
food
blood.
coloring. It does look like blood. And I think I put too much, so I'm just trying to smear it out a little bit.
Here's the cover.
Maybe now I'll put a cover on. And these are called slide covers?
Cover.
Slide covers, awesome.
Tops.
I like to call them tops.
Tops.
Alright,
Top,
let's
top.
see if I can
Tops.
get anything here.
Oh, there it was. Move down a little bit. This way, towards you.
Towards me?
There we go.
What? Look at that. So I actually was able to dye Robert's cheek cells just using food coloring. You don't need any fancy methyl blue to actually do that. Now, maybe the way it dyes the cell might be different because methyl blue is supposed to dye the nucleus, right?
Methylene, yes.
Methylene blue. But I'm pretty sure that we could say that Robert is officially alive. Robert, look at you. You're gonna do your dance. You were right. You're alive. That's it. That was a very exciting dance. It was like, yay.
Okay, so Star Sonic said, okay, now check the rest of everyone else on Earth just to be sure Robert's not the only one up.
Yep, that sounds like a good experiment. Robert, you now have to go check cheek cells from everybody and
Now
see
let's
if
do
they're
it
alive.
to you.
You know what? Another good follow-up that we thought would be cool for this is if we were to look at the cheek cells of a different animal.
Cool.
So we were thinking about doing that with Cole. I don't know if we have enough time.
Why not Penny? She's right there.
Oh, Penny's easier. Can we get Penny? Let's see.
Okay.
She's looking at me like, oh my gosh.
Like, what are you gonna
I
do?
did it, Penny.
All right, so we have our secret object today that we could do another test on here. And this is our dog and a clean Q-tip.
I have one.
Oh, you do? I said Q-tip. I meant toothpick. So this is our little Chihuahua.
Tampon for your
And we've done this before to send off her DNA, so we know that she has dog DNA. Well, let's see if we can do this. Come here, Pen. You be a good girl. She's licking it. She's having a good old time. Awesome. Clean slide.
Yes.
Now, while I'm prepping this slide real quick, I could mention that if you look at Robert's cells that you have under there, you could actually see some stuff that's in there that adds different colors. So some stuff is really dark, some stuff is lighter. And if you look, there's a little tiny point, usually somewhere in each cell. I wonder what that is. All right, wonder what that is. Should I put the dye on here? Let's see if I can see what that. I don't know if I got anything. It's kind of hard. Harder than doing it on Robert. I don't see anything. Maybe I didn't get any cells.
Yep. There we go. There's a bunch of stuff in there. Penny, you're alive.
So it looks like maybe our dog is alive too. We found some cells.
Robert, come. Come see what we found in Penny.
Oh, they look cute.
Yeah, they're cute. So these are our dog cells compared to human cells. And they don't look too different, right? I wonder if the difference between human cells and dog cells are something that's inside of the cells. What do you think, Robert? I'm gonna dye them really quickly. I know I'm a little bit over time, but hey, when you're having fun, what's a couple minutes? Awesome. Again, I'm just using food coloring as a dye. I don't know if this is lab approved, but it is approved for us and our discovery. Uh-oh.